My name is Eric Speckin. I work for Speckin Forensic Laboratories in Okemos, Michigan. My primary job is to determine when documents were written and how long ink has been on paper. For instance, if I were contacted on a case to determine if a document was written in April of 1996 or later, I would first determine what type of ink is on the document. Then I would compare that to a known dated standard to determine if it matched an April 96 standard or a later standard. This is because inks take approximately 3 to 3.5 years to dry completely on paper. Before an ink is 3.5 years old, it will undergo a drying process. You can then compare that to other standards to determine where in the drying process a questioned ink is. If I identified that the ink was a Bic black, I could retrieve a document from my known library of inks and compare the April 96 portion of my library to the April 97 portion and determine if it was written in April 96 or later. In order to do that, I would need several tools first. The first step would be to use a punch and a backer. I would place the document on the backer, which is a self-healing board. I would then take and punch a micro plug of ink from the document, place it into a vial, and plunge the plug into the vial. I would put approximately 6 plugs per vial. And I would run the test in duplicate. So I would have 2 plugs from the April 96, 2 plugs from April 97, or 2 vials, and 2 vials of my question entry. I would then measure the dryness level of the ink using 2 separate methods. The first is using a weak solvent to determine the rate of extraction from the paper. This is done by taking a syringe like this and a weak solvent from a vial and injecting a fixed volume, in this case 16 microliters, into the vial. And taking aliquots of ink at desired time intervals using a volumetric pipette. The first sample would be taken at 30 seconds, the next sample at 1.5 minutes, and the following sample at 3 minutes. More samples can be taken if desired. At 30 seconds, the solution is mixed, the pipette is inserted, it fills automatically. It's then removed with tweezers. And spotted on a glass-backed silica gel TLC plate. Until the entire volume has been placed onto the plate. Then at 1.5 minutes, the second sample is taken in the same way the first was taken. And again, the entire volume is spotted. And for purposes of this demonstration, we will take one more sample at 3 minutes. Once all the desired volumes or samples have been taken using the weak solvent from all the vials placed along the bottom of the TLC plate, the second method is to use the percent of extraction. The way this is done is the volume of weak solvent that is still left in the vials is then dried in an oven and removed from the sample completely. Before the samples are placed in the oven to be dried, the temperature is read on the thermometer and a fine adjustment is made using the temperature control to turn it either up or down to obtain the desired 80 degrees Celsius to dry the weak solvent from the samples. Once the oven has maintained 80 degrees Celsius, the samples are then placed in the vial rack into the oven for approximately 30 minutes to dry the weak solvent from the plugs. After the 30 minutes, the vials are then removed from the oven and allowed to cool before the strong solvent is added. Once the samples have been taken out of the oven, they're allowed to cool, and once they have cooled, we will inject a strong solvent, in this case benzyl alcohol, into the vials and we'll use a smaller volume since we're only taking one aliquot or one sample to measure. In this case, I've used 10 microliters. I would inject it into each vial that I have a sample in, and using a timer in the same way that I measured the rate of extractions times, allow it to extract for 10 minutes. Once the 10 minutes is up, the sample is then rotated and mixed the same as the rate of extraction. And the strong solvent spot is placed directly after the series of weak solvent spots that measure the rate of extraction. Once all the spots have been placed from each vial on the plate. The plate is then placed into the same oven, and the spots are completely dried. After all the strong solvent spots have been placed on the plate is then put in the oven at 80 degrees Celsius for approximately 30 minutes to completely dry all the solvent from the plate and from the silica gel on the plate. This is a Uniscan densitometer. It's a video scanning densitometer, and it's designed to determine how much light passes through an object or how dense in color an object or a sample is. It contains a white light box, which is this here, with a transfuser which divides the light up evenly throughout the inside of the box. The plate, the TLC plate, is then placed on top of the diffuser. The light passes through the plate into the camera lens, which is here, which is zoomed in on the object, and the f-stop controls how much light is allowed to pass into the camera. The camera contains two outputs. One is to the monitor, which will display the samples and the lanes that are to be scanned. The second output is to the computer, which has computerized integration software to allow the computer to determine how much light is passing through the sample, and then displayed on the computer screen. Once the plate is removed and cooled from the oven, it is placed inside and aligned on the monitor, and then zoomed in to be as large as possible and still include all four spots. The first spot we see is the 30-second extraction. The second is the 90-second extraction. The third is the 3-minute. And the faint spot on the end, or the fourth spot, is the strong solvent extraction. In this case, most of the ink had been extracted in a weak solvent, and not much was left for the strong solvent to extract. The relative densities are then measured using the computer. In order to set the densitometer to make sure it's working properly before we scan, we pick digitizer set from the scan menu. There are two parameters that we need to make sure are correct. The first is that the baseline is not flat, and the second is that at the strongest absorbance that the peak does not go off the screen. So we will pick the strongest spot and travel completely across to make sure that it never leaves the screen, and we can see that it never does. Also, the baseline never becomes flat and touches zero. So the densitometer is set properly and the digitizer is working. Once the sample is then adjusted, we go to the scan menu and parameters, and we set the parameters. In this case, we're going to scan 4 lanes. We will make 20 passes, and then we pick set to set the edge boundaries of each lane. It will measure 20 passes and take the average for each lane that is measured. So in viewing the monitor, we will set the edge of the lane of the first spot, the left edge, at 34, and the right edge will be set at 52. The second spot, the left edge at 94, the right edge at 112. The third spot, the left edge and the right edge. The final spot is then larger than the previous 3. And we set that. We can then click on View, and on the monitor it will display the lanes that are going to be scanned. If those are correct, we click OK. We then click again on the scan menu and go to baseline. We will scan 2 empty lanes, 20 passes each, and click OK. This will scan 2 lanes on either side of the sample and subtract off any background information from each sample to allow only the ink to be measured. We will then scan the samples by clicking on Scan. Do one more thing. Once the densitometer has finished scanning, the peaks will appear on the screen for each lane that was scanned. The more red that appears, or the bigger the peak, the more ink that's present and the more ink that has been extracted from the paper. We can see as we go up in extraction time from the 30-second to the 90-second, the peak gets larger and more ink has extracted. And again at the 3-minute, and this is the total extraction, which we can see is less. This scanning procedure and calculations are done for all samples. The samples are then intercompared. If there is a difference between the 96 and the 97 sample, it can be determined then which sample the question matches and therefore when the writing has occurred, if it was done in 96 or later.